हेलो, गुड इवनिंग। वॉइस सही है क्या? इज इट क्लियर? Hello, good evening, good evening. Is my voice is clear? Can anyone confirm? Patel? Annu? Is my voice is clear? Yeah, okay. Yeah, yes, yes. Clear, clear. ठीक है, थोड़ा जल्दी फटाफट शेयर कर दो सबको, आपका फ्रेंड्स के साथ ग्रुप्स में, सारे कौन कौन प्रिपेयर हो रहा है प्लांट साइंस में, सबको भेज दो, ठीक है। ये तो रिसेंट पेपर है लास्ट ईयर का। वी विल डिस्कस टुडे ICAR PG 2025 paper. In that we will discuss plant breeding section. Okay, again there will be so many questions which are concentrated on heterosis breeding in this particular paper, that is hybrid breeding. Okay, we will see what are the questions, and also someone asked, I have left one question from previous paper. Okay, luckily someone kept in the group, so I will start with that particular question. Okay, the last year missed question. Nutan, I sent it, sir. Good, good, Nutan. ओके, भेज दो बस। 30 दिखा रहा है, आप सबको शेयर किया है नहीं किया है सारे लोग? अरे सर, ये तो बस हम के लिए, हम नहीं शेयर करेंगे। ऐसे ऐसी भी है क्या? ओके, मुझे पता है सारे लाइव होने के बाद भी मतलब नॉर्मल वीडियो भी देखते हैं लोग, मतलब लाइव में थोड़ा मजा आता है ना, हम सबके साथ इंटरेक्शन कर सकते हैं, ठीक है, कमेंट में ऐसे। ओके देन, share is care. Yeah, yes, Nutan, good. Okay, then we will start today's session with the previous year paper. That means in previous year I have left one question. I don't know how I left this important question which is related to gamete selection. Okay, the question is saying which of the following combination of the premises of gametic selection in a plant breeding is the correct one? बताओ, आंसर बताओ। They are asking about which are the following statements are true regarding gamete selection. Multi-parent crosses are required for simultaneous improvements of multiple traits. The male parents of the final crosses are heterozygous and hence heterogametic. Each zygotic seed is a product of separate independent fertilization event, and gametic selection is an effective method to isolate for inbreed for hybrid breeding. इस चार स्टेटमेंट में बताओ कौन सा एक गलत है इसमें, ठीक है, बस वो आइडेंटिफाई करना पड़ेगा। बताओ आंसर। सी सी सी, A C D ओनली। अरे भाई सी कैसे होगा? You are okay, you are identifying the wrong statement or you are telling the C option? Okay, D. Okay, before that, before choosing the correct answer, we will discuss what exactly the gamete selection. So we will come back to this particular question later. What exactly the gametic selection? We will see the procedure, so that we will see. First we will start with inbred. Okay, what we are doing? We are doing the inbred improvement here. Okay, we will cross our particular inbred into OPV. Your OPV contains number of heterozygous and heterogametic individuals. Discuss ಮಾಡ್ತೀವಿ 2025 ಪೇಪರ್ ತಾನೆ 2024 ತಕೊಂಡಿಲ್ಲ ಮಿಸ್ ಹಾಕೊಂಡಿಲ್ಲ ಫಸ್ಟ್ ಕ್ವೆಶ್ಚನ್ ಅದು ಮಿಸ್ ಆಗಿತ್ತು ಲಾಸ್ಟ್ ಇಯರ್ ಅಂತ ಹೇಳಿದ್ನಲ್ಲ. Okay, inbred into OPV. So after this you will get F1 plants. Okay, here each F1 individuals, okay, each F1 individuals are very different, since your inbred, okay, is crossing with OPV. Each OPV is having different alleles at that particular loci. That's why they are heterozygous and heterogeneous. So whatever F1 you will get in this particular stage, again these are completely different individuals, not exactly like inbred 1 and inbred 2. When you will cross inbred 1 into inbred 2, the F1 you are getting are homogeneous. But in this case, okay, since you are crossing with OPV, OPV will provide, each OPV will provide different gametes, okay, gametic combination. So F1 also, each F1 also very different. Okay, so what you will do, then you will, each F1 you will cross with any tester. Okay, your tester should be broad genetic base. Then you will get progenies, the crossed progenies. Okay, and you will also go for selfing of this particular F1 plants. Okay, selfing, simultaneous selfing of this particular F1 plants, and also you are crossing this F1 plants with the tester. Okay, you will evaluate these progenies. Based on that, the whichever progenies they are giving good performance, okay, by using those selfed seeds you will go for next season. Okay, these are the selfed progenies which are giving best performance in the when you are crossed with the testers. Okay, so that again you will go for F2, then you will choose the best individuals, and again you will go for F3. Like this till you will go from F6 to F7. Okay, then your inbred version after improvement. Okay, inbred after improvement. Why we are doing it? Okay, since the name itself suggests gametic selection, we are selecting gametes here, not zygotes. You can see here OPV is providing male gamete. Okay, and inbred it contains in their genomic level it contains similar homogeneous condition. When you are crossing with OPV, your each F1 is having different male gametes, so you are selecting the superior gametes. Okay, in gamete selection, that's why itself suggests you are selecting the gametes. Okay, not zygotes. You are selecting P, not P square. Okay, the superior allele is your P. Okay, your RD Weinberg also, your P will tells about the superior, any matlab A or small a, but we will take P as superior allele. Okay, we are selecting superior allele rather than superior homozygotes or P square. Okay, here we are choosing the superior male gametes from the test class progenies. Then out of these, we will go for, anyway we are going for selfing, we'll select the best improved version of inbred. Okay, we are accumulating the favorable allele here. Again, once again, it is nothing but your recurrent selection, but it's based on your gametes, not zygotes. Okay, we are not testing the zygotes here. You are, yes, you are testing the F1 plants, but each F1 plants is different since your OPV is giving the different gametes. Okay, that's the basis of your gametic selection. Okay, we can come here now. Multi-parent crosses are required for simultaneous improvements of multiple traits. Yes, at particular time, okay, based on your interest, your OPV contains different traits. Okay, like that, if you want to improve for multiple traits, then you should target the multiple crosses and multiple traits. The second statement, the male parents of the phylum crosses are heterozygous and hence heterogametic. You can see the male parent here inbred into OPV. Your OPV is heterozygous and heterogametic. Okay, whenever you will repeat this cycle again, your male parent will be OPV only. All other are, you can see, the selfing. In selfing also, these are heterozygous and heterogametic. Okay, your statement 2 is correct. Each zygotic seed is a product of separate independent fertilization event. Each zygotic seed, okay, you can see here, is separate independent event. Okay, your one gamete will be similar, which is comes from inbred, and another gamete from OPV. You can see different OPV will produce different male gamete. So each zygotic seed will be product of different events. वो भी सही है, ठीक है. Then what about the last one? Gametic selection is an effective method to isolate inbred for hybrid breeding. Is it correct? About last statement, if it is not correct, then how you will make this correct? How you can correct this statement? बताओ कोई. Gametic selection is an effective method to isolate an inbred for hybrid breeding. You can just see the flow here. We are starting with inbred and we are ending with improvement of inbred. We are not isolating any inbred here. Okay, we are starting with inbred only. Okay, your gametic selection is an effective method to improvement of an inbred. Yes, good Bhavesh. It is an improvement method. Okay, we are not easily isolating, and also it is not an effective method for isolation of inbred also. Okay, since we are not sure about your gametes sometimes. Okay, and it's not an effective method to isolate those superior gametes. Okay, this is the condition. So the answer will be A, B, C are the correct answer. Clear? बताओ, कमेंट में बताओ, कुछ डाउट्स है क्या? Okay, good. This is about the last year paper, the last year question, one we missed in 2024 question missed in the last year discussion. So now we will move into our the main paper, ICAR PG exam 2025, this last year paper. Okay, we'll start with the particular question. In the context of ex situ conservation of plant genetic resources, what is the primary benefit of integrating molecular markers technologies during germplasm characterization and evaluation? They are asking about molecular marker technologies. What are the benefits of molecular marker technologies in case of your germplasm conservation? B. They enable accurate and efficient evaluation of genetic diversity and help in identify genetically redundant accession. The thing is, okay, if you use the molecular markers, okay, you can avoid the duplicates. Okay, you are having some germplasms, you are collected from some area. Okay, without knowing you, okay, again you will collect the same germplasm from another place. If both are the similar germplasms, okay, at molecular level, then what's the need to conserve the both of germplasms? Okay, again, in germplasm conservation, the space will be ultimate thing. Okay, you should minimize the number of accessions. Okay, based on duplicates, you can avoid by using molecular markers. Okay, anyway, you know already what exactly the markers are. Okay, I have already discussed about it. So, and it will enable the accurate and efficient evaluation. Okay, accurate and efficient is nothing but, okay, the minimum germplasms. Okay, you can evaluate and analyze the genetic diversity. Okay, one of the other application of your markers in case of conservation to analyze the genetic diversity at molecular level, so that you can use those germplasms in your breeding pipeline. Certain plant species produce recalcitrant seeds that are highly sensitive to desiccation and low temperature, making traditional seed banking unsuitable. Which of the following in situ conservation approaches is most appropriate for long-term preservation of such species? Recalcitrant seeds. Okay, if you reduce the moisture of recalcitrant seeds, then your viability will reduce. And if you store those seeds in low temperature, then again your viability will lose. Okay, there are so many limitations with recalcitrant seeds. Then which of the methods we can go? A sir, C, A, C, A, C, confusion हो रहा है C में, ठीक है? In vitro tissue culture combined with cryopreservation. Okay, as it circumvents the drying intolerance. Anyway, you know, if you go for drying of recalcitrant seeds, if you lose the moisture, then again there will be loss of viability. So to avoid that, okay, you can go for in vitro tissue culture. Okay, in vitro tissue culture is nothing but again, okay, based on meristem culture, based on different organs culture. Okay, you can go for this particular tissue culture methods with the cryopreservation. Okay, this is the best method to avoid the desiccation intolerance. You can see here, some are telling about A, establishing field gene banks because natural conditions are best replicated in situ. Sometimes I will agree for field gene banks, but the problem is, okay, if you go for field gene banks, you are giving environment to that particular germplasms. Okay, there may be again changes in at genetic level. There will be some mutations. Okay, there will be some aberrations due to the growing period. Okay, there will be some variation if you go for field gene banks. But if you want to conserve as it is, then in vitro tissue culture is best with the cryopreservation. Yes, we can also observe here somoclonal variation, but compared to field gene bank, again in field gene bank you need it is laborious, then you should be having the high space. Okay, to avoid that, you will go for in vitro culture. Clear? क्लियर है ना बात? बताओ. Next question. Which statement is generally true regarding BT cotton hybrids in India? They are asking about BT cotton. Yes sir, yes sir. B. नूतन का इमोजी आ गया अभी, कोई नया वाला. ठीक है, B sir, B. They incorporate genes for resistance against bollworm complex. Yes. Your BT cotton, the chemical which it produces, the gene, okay, the cry protein is effectively against the bollworm complex. So there will be a 3 different generation of your BT cotton, that is BT1, BT2 and triple gene hybrids. Okay, in 2002 the event is released, okay, in commercial cultivation in India, that is Volgard 1 only with cry AC gene. Okay, in BT cotton, that is Volgard 2 cry 1 AC with cry 2 AB. Now recently in from last 5-6 years, okay, the triple gene hybrids, okay, cry 1 AC, cry 2 AB and third gene WIP 3A. WIP 3A is nothing but your cry 1 AC and cry 2 AB, they will work on the particular growth stage of that particular larva. okay, at larval stage. That too during that, when that alkaline pH, okay, the gut epithelial cell, when they will show the alkaline pH, your cry protein will work during that particular larval stage. But in case of your third gene, that is VIP3A, okay, it will work on all vegetative stages of that particular larval growth. Okay, this is all about the 3 generation BT cotton. So that's why your second statement will be correct. Again, there will be a question on BT cotton. You can see here. The Bacillus thuringiensis, a soil bacterium, possesses Cry1AC gene that kills larva of bollworm, and the reason Cry1AC gene codes for crystal protein, which is responsible for the insecticidal effect. मैं तो डिस्कस अभी भी किया था पिछले स्लाइड में, ठीक है? ये yes, your both A and R are true, and R is the correct explanation of A. Your Cry1AC gene, Cry2AB gene, both will produce cry protein. Okay, that cry protein is toxic when your insect gut epithelium is alkaline pH. Okay, that another thing I want to tell here. Okay, if those leaves, if they got feedden by your any cows or any humans, okay, it will not affect that cry protein, will not affect that particular organisms. Okay, why? The insect, particularly the insect gut, is in alkaline condition. But in humans, okay, in case of humans or the other organisms, the gut epithelium, okay, the pH condition will be acidic condition. At that particular pH, okay, the cry protein will not toxic, but if it is alkaline condition, your cry protein will be in the toxic condition. Okay, that's why your first option will be correct answer. Okay, it is a correct, clearly explaining reason for your assertion. ठीक है?
ठीक है, ठीक है. So hi everyone, sorry to interrupt करने के लिए sorry. मैं भी सुन रहा था क्लास. ठीक है, मैं भी बोल रहा हूँ. तो yeah, so why I'm here just to discuss about the upcoming plan. Next, इसके बाद आपको क्या चाहिए? Like you want the PYQs of genetics and seed science first और marathon? तुम लोगों की पूछ लेता हूँ. मेरा गेटअप भी चेंज हो गया, इसका मतलब भी आप समझ गए होंगे कि क्यों चेंज हो गया. Yes, good evening everyone. तो seed science न्यू लुक. सो आपके पास ऑप्शंस रख रहा हूँ कि आपको नेक्स्ट क्या चाहिए. लाइक कल ही आएगा, बहुत लेट नहीं, कल ही. ऐसा नहीं कि आज बोल के जा रहा हूँ और 2 दिन बाद, नहीं, कल ही लेके आएंगे. सो मंडे से मेरा, मंडे तो हो गया ना आज, ठीक है, वेडनेसडे से मेरा 2 क्लास बचे हुए हैं, तो उसका क्लासेस करा रहा हूँ. बट कल के लिए कुछ बता दो कि tomorrow shall we complete this genetics 2025 PYQs and seed science PYQs and then start the marathon. Seed book has arrived. Acha, good, fine. Genetics marathon, seed science marathon. अच्छा, like tell me how many of you already analyzed the 2025 पेपर को already solve कितने लोगों ने किया है? I guess इसीलिए बोल रहे हो कि नहीं चाहिए पेपर करके. If you have done it, thumbs up. If you have done already the PYQ analysis of plant sciences 2025. Yes. अच्छा, good. Yes sir, PYQs first. Okay, seed science मक्का marathon. Okay, सब कुछ खत्म हो जाएगा. And we are what we are planning. Like let me just highlight the plan. कि plan पता होना भी जरूरी है कि आपको पता चले कि हम कब तक क्या कर रहे हैं, what we are doing. So as of now we plan कि जो प्लांट पैथोलॉजी मेन था उसका मैराथन कंप्लीट है, PYQs कंप्लीट है. नेक्स्ट नाउ वी आर कंप्लीटिंग द प्लांट ब्रीडिंग. आज के बाद प्लांट ब्रीडिंग भी कंप्लीट है. तो बच क्या रहा है? बच सिर्फ सीड साइंस और जेनेटिक्स हो रहा है. तो उसका मैं पहले 2025 का PYQs खत्म करता हूँ. उसके बाद आई विल नॉट गो टू द 2023, 2022, 2021 नाउ. फर्स्ट आई कंप्लीट द मैराथन, आई कंप्लीट बोथ द जेनेटिक्स एंड द सीड साइंस मैराथन. उसके बाद टाइम बच रहा है, देन वील गो फॉर द PYQs ऑफ द अदर parts like genetics and seed science of later years, because already वो चीज़ है YouTube में, but स्मार्ट बोर्ड पे नहीं है. तो जो चीज़ नहीं है उसका पहले प्रायोरिटी देते हैं, उसके बाद उसका कर लेंगे. ठीक है. सो आई वांट इट सर, PYQs एनालाइज मैराथन. ठीक है, सब मिल जाएगा भाई, सब कुछ मिलेगा, एवरीथिंग. देखो कि इस वीडियो में मुझे एटलीस्ट 100 कमेंट्स चाहिए आपके. मैं तो भूखा हूँ उस कमेंट्स का. I need the comments from you people. Like after watching this video, like if you want the marathon series of genetics and seed science, just comment on seed science marathon. ज़्यादा नहीं टाइम इन्वेस्ट करना है कमेंट पे, पढ़ना है ज़्यादा से ज़्यादा, जस्ट सिर्फ एक कमेंट करना है और इसे लास्ट सेंड ऑफ दे दो. मैं अभी PYQs के लिए जा रहा है अभिषेक, तो आएगा फिर से ब्रीडिंग मैराथन में, बट PYQs खत्म करके आज अभी जा रहा है. Okay, I need like more and more interaction. I need more and more response from you people. Like, this is a peak time. I told you already that you have to be very thorough and you should be consistent in this last 20-25 days which is left. You should be more energetic. थोड़ा energy saving mode में भी रहना है, अच्छे से नींद भी करना है. But yes, your brain should be always active. Last एक महीने का टाइम है, एक महीना ही बोलूंगा मैं इसको, क्योंकि लगभग ऐसा ही मान के चलो कि आपके पास एक महीना है अभी करने के लिए, और करो अच्छे से. You will do it. Please share the PDF of plant pathology. Plant pathology का PDF already uploaded है, marathon series का PDF test series course में uploaded है. Please go and access it. When will the marathon start? See, just after completing this class, tomorrow I will be completing genetics का 2025 PYQs, and if possible tomorrow only I will be completing the seed science PYQs 2025, and in the next day hopefully the plant breeding, हाँ plant breeding marathon चलेगा परसों से.
हाँ चलेगा,
परसों से plant breeding marathon चलेगा. कितना क्लास करने वाले हो?
3 क्लास मैराथन में, 3 क्लास में हम सारे ब्रीडिंग का पोर्शन कवर करेंगे. ठीक है.
So in which parts, like
First will be your the principles, basic principles of your plant breeding. That will include your male sterility and self compatibility. Second one is breeding method for self pollinated and cross pollinated crop plants. Then third will be your special breeding methods,
okay,
which includes your IPR and everything, the remaining portion.
Okay, देखो solid में 3 क्लासेस में प्लान ब्रीडिंग का खत्म हो जाएगा, तो 3 क्लासेस बिल्कुल मिस नहीं करना है आपको. जो क्लासेस हो रहा है मैराथन उसको अच्छे से देखना है, चाहे आप मास्टर कोर्स में सब कुछ देखा हो या नहीं देखा हो, फिर भी आप सबको देखना है चीज. एंड नेक्स्ट यू हैव टू आल्सो एक्सेस द पीडीएफ्स एंड गो थ्रू इट वंस. ये पीडीएफ बना क्लास के लिए प्रिपेयर करना जितना टाइम नहीं जाता है, पीडीएफ बनाने में चला जाता है. लेकिन सो मच ऑफ टाइम नीड टू इन्वेस्ट ऑन दिस पीडीएफ प्रॉपर बनाने में. इधर उधर से उठा के ऐसा नहीं फेंका जा सकता है इसमें. पीडीएफ में बहुत अच्छे से बनाना पड़ता है. इट्स टाइम टेकिंग प्रोसेस, उसका फायदा उठा लेना एंड मेक इट गुड यूज ऑफ इट. सो एज सून एज ही कंप्लीट्स द मैराथन ऑफ प्लांट ब्रीडिंग, उसके एक दिन बाद मैं जेनेटिक्स का स्टार्ट कर ही दूंगा. ठीक है? इज इट ओके? एक्साइटेड? ओके. एडिटेड या एक्साइटेड? हाँ एक्साइटेड. ठीक है, ओके. चलो ठीक है, कंटिन्यू कर लो. सिर्फ मुझे इन्फॉर्म करना था ताकि आप लोग अपना शेड्यूल को ऐसे प्रॉपरली मेंटेन करो और थोड़ा गैप रखो कि मैराथन के लिए, पीवाईक्यूज के लिए. कल मैं सुबह 10 बजे आ जाऊंगा. आई विल बी देयर इन morning 10 AM with the genetics PYQs. So hope so I'll meet you all again after a very long time. Where can I find the marathon PDF? Marathon PDF तो मैंने बता तो दिया.
Yes, mock test bundle में
एक बार दिखाई दो भाई. See, if you are opening the website and if you are opening the courses जो आपने टेस्ट सीरीज ले रखा होगा, see, everyone might have purchased this. If you are a student of master course already, this course might be already visible in your course dashboard or library. जो if you scroll down, ये जो course content में scroll करोगे आप नीचे तो यहाँ पे after this schedule mock test you will get the plans and test series. Okay, PYQs. Okay, marathon series देख रहे हो यहाँ पे. Everyone can see this. ICAR PG Marathon Series, ICAR PG 2026 Plant Pathology Marathon, complete rapid revision class 1, class 2, class 3. So under this, like you can find the class 1 PDF, class 2 PDF, class 3 PDF. Everything is available here. सब कुछ है यहाँ पे.
एक स्टूडेंट बता रहे हैं, सर प्लीज सर आई हैव नॉट परचेज टेस्ट सीरीज.
अरे भाई
जाके परचेज करो, अभी ज्यादा मिलेगा इंफॉर्मेशन.
See, it is just for 399. 399 में ही है ना, ज्यादा तो नहीं बोल रहा हूँ मैं. Yes, it is available just 399. What else can I tell you to do?
पूजा ICAR सही से बता रहा है. Any one course you can enroll, then you will get all agreed PDF.
Yes,
definitely.
See, we are just asking you to enroll for a 399 course. It has like more than 150 plus mock tests. और आपके पास भी टाइम है, ऐसा नहीं कि मैं लास्ट दिन पहले आपको बोल रहा हूँ कि कोर्स परचेस कर लो. मैं मास्टर कोर्स के बारे में बिल्कुल बात नहीं कर रहा हूँ, छोड़ ही दिया था मैंने बात करना एक महीने पहले ही. नाउ आई एम टॉकिंग अबाउट ओनली द टेस्ट सीरीज. टेस्ट सीरीज विल डेफिनेटली हेल्प यू. वो लेना भी होगा, क्योंकि अगर आपको दिल से अगर एग्जाम को क्रैक करना है तो मोर एंड मोर क्वेश्चंस को आपको अटेंड करना होगा. एंड ऑल्सो अटेंडिंग एंड like making the questions or attending those questions which are quality standard, वो भी बहुत important है. ऐसा कचरा आप अटेंड करते रहोगे तो कॉन्फिडेंस लो हो जाएगा. So this test series is not an effort of a single year. It is the effort since 2021. 2021 से हम efforts डालते आ रहे हैं, तभी ये mock test series बना है. सो इससे ज़्यादा मैं क्या ही बोलूँगा. So please make the classes on EO. EO के लिए एक क्रैश कोर्स निकालने का कोशिश कर रहे हैं. So let me I will update about it later. Okay. So please make a mock test for agriculture officer. ठीक है. Okay. Okay guys. So this is my information कि कल genetics का marathon होगा and also like if possible seed science का हो जाएगा and that completes the PYQ series currently and next day after tomorrow you'll be starting the plant breeding marathon. 3 दिन solid चलेगा. उसके बाद मैं आके genetics marathon start कर दूंगा. Be ready for it. Okay.
Okay, we will move to our next question. The simple question. Okay, we can. दयान रिट्रैक्ट कर दिया कुछ मैसेज. साजन टाइम फॉर मैराथन. हाँ मतलब मेरा मैराथन चाहिए तो आपका ना इस लास्ट वीडियो मतलब एटलीस्ट 2025 का वीडियो है ना वो 2K व्यूज होना चाहिए. ठीक है, उसके बाद ही मैं आएगा. ठीक है, आपका भी आपका मर्जी कितना शेयर कर सकते उतना करो. 2K बना दो लास्ट ये 2025 पेपर का वीडियो. ओके, देन वी विल स्टार्ट मैराथन. अदरवाइज आई विल नॉट कम. पिजन पी is considered as an often cross planted crop. वो सबको पता है. पिजन पी का मतलब often cross planted crop आप लिस्ट बना के रखा दिया ऑलरेडी. ओके, your pigeon pea, your cotton, okay, sorghum, okay, these are often bajra, often cross planted crop plants. Okay, in pigeon pea they are particularly talking about pigeon pea. Although self pollination can occur within the bud before the flower opens, cross pollination 20 to 70% often facilitated by the insects like bees is a common and significant factor in seed set. That is the reason they are telling why it is considered as cross planted, often cross planted crop plants. Your answer will be A. Both A and R are the true and R is the correct explanation of your assertion. A plant with both staminate and pistillate flowers are separate branches of the same plant are the best described as which of the condition, flower condition. Okay, ये तो सीड साइंस में भी आएगा. मैं ऐसे उठा के इधर लगा दिया. ठीक है, which one is you can see both staminate and pistillate in the same plant but in the different branches. For best example is your maize plant. Okay, there will be a tassel and there will be a cob नीचे. ठीक है, it will contains the both the flowers in the different branches. It is your monoecious. Okay, the monoecious plant. Dioecious is nothing but it will only contains any one of the sex. Maybe the staminate flowers or pistillate flowers. The best example is your date palm. Okay, and anyway, apomictic, those are the different seeds. Those are like a sexually reproduced seeds. It is not a without sexual reproduction. Okay, your clistogamous. Any examples for clistogamous? Yeah, papaya also. It will comes in dioecious. Good. Clistogamous. Anyone? Your flower opens after pollination. Gaetogamy is a method of monoecious condition. Yeah, the gaetogamy is nothing but the sexual reproduction method that you can see in monoecious. Okay, the pollen of same plant will place on the same plant of different flower. That is your gaetogamy. Yes, wheat, rice, pigeon pea, rice, wheat. Yes, your rice and wheat are good example. Okay, which is not an multi-parent mapping population. Not an multi-parent mapping population. पहले आपको पता होना चाहिए मैपिंग पॉपुलेशन क्या होता है. ठीक है, व्हाट इज एग्जैक्टली द मैपिंग पॉपुलेशन. ओके, इट इज नथिंग बट द पॉपुलेशन. पॉपुलेशन तो आपको पता है, मतलब your F2 population, F3 population, okay, your nils, near isogenic lines, and your recombinant inbred lines. Okay, these are the different mapping population. Those are nothing but populations, but we know the segregation ratio of that particular population. That's why we'll call it as a mapping population. Okay, we know how the segregation is happening. In case of F2, you know 1:2:1. Okay, you know this ratio, right? In case of your nils and rils, 1:1, 1:1. That means your rils, okay, your recombinant inbred lines or your nils, they contain capital AA or small aa. But your F2 contains capital AA, capital A, small a, and small a, small a. Okay, we know the segregation ratio. That's why we'll call that particular population as a mapping population, so that we can use those population to locate the genes, okay, to map the genes. This is the basic idea. At this point of time, you should know. They are asking which is not a multi-parent mapping population. There will be two types, biparent and multi-parent. When you are developing this particular population, you will use parents. If you are using only two, in all of these cases, you will only use only two parents. You will start with two parents. Okay, that's why you will call it as a biparental mapping population. But in some of the population, you will go more than two parents. Okay, those are your magic. Okay, multi-parent advanced generation intercrosses. Your NAM, nested association mapping. In this case also, you will use more number of parents. Okay, diverse parents. And your association mapping panel, the panel itself will tells that it contains the different number of, the more number of germplasms. But your F2 derived F3 plants, okay, your F2 derived F3 will comes under biparental mapping population. That means only you will use two parents. Okay, कौन कौन लगा रहा है D? Bhavesh, co-segregation of marker and trait. Yes, good. Anyway, you are MSc students, okay, you are preparing for PhD, so that you should know the basic principle of your mapping is co-segregation of marker and trait. But anyway, for UG students, okay, they should know that you know the segregation ratio of population, so that you can call it as a mapping population, since we can use this particular population to map the genes. Okay? In association mapping, sir, natural population takes care of it. Yes, good. Okay, association mapping is nothing but, mapping panel is nothing but the germplasm. Okay, the number of germplasms of that particular crop. Okay, anyway, it also contains again the more number of parents. Okay, this is the thing. If I want to explain bit more further how we can develop mapping populations, you can see P1 into P2. You will get F1, then you will get F2. This is one kind of population, okay, F2 population. Then from each F2, if you use the single seed descent method, from each F2 plants, you will take only one seed and you will go for next up to F6 using SSD. Then you will call them as rils, recombinant inbred lines. Okay, this is the one kind of population, again rils. Then if you use your F1, if you cross with any one of the parent, you will get PC1F1. This is backcross population again. Okay, one of the population. Then if you continue this backcross, okay, till BC6 F1, then you will go for selfing BC6 F6. Then you will call this as a NILs, near isogenic lines. This is again one part of the population, mapping population. Again, you can see here, if you take this F1, okay, you are taking this F1 and doubling of chromosome. Okay, your DH population, double haploids. Are you getting it? Okay, how we can develop different types of populations. Okay, double haploids, F2, F6, that is your rils, your nils, your backcross population. But another one kinds of population, okay, that you have seen in your answer. Here from each F2 plants, there will be 2000 F2 plants. From each F2 plants, if you are selecting one seed and you are bulking it for the next generation till F6, then you will call those as a rils, recombinant inbred lines. But if you grow each F2 plants in separate progenies, okay, there will be three kinds of genotypes in F2. If you grow those F2 plants in a single separate row, okay, of particular F2. Okay, your F2 contains capital A, capital A, and small a. Then you will grow those particular genotype into different rows, separate rows. Okay, then you will call this as a F2 derived F3. Okay, F2 derived F3. Okay, these are the different kinds of population. Okay, at this particular point of time, we should know the basics. Okay, how we will produce different mapping populations. Okay, these are all your biparental mapping population. But in case of multi-parent, first one is your magic. Okay, multi-parent advanced generation intercross. How we will develop? You will select some parents. Okay, some 8-10 parents. I will take 8 parents, P1 to P2, P3 into P4, P5 into P6, P7 into P8. These are different parents, multi-parents. Then you will get F1, 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 F1. Then you will go for all possible crosses between different F1s. Then again you will get F1 here. Okay. Again you will get F1 here. Then you will ultimately will cross. Then again you will get F1. Then you will go for again continuous selfing. Then you will get mapping magic population. Okay. First one, all possible double crosses. Then all possible three-way crosses, all possible crosses. Okay. At the end of the day, you will get magic population. In case of your NAM, that is nested associated mapping. Okay, there will be one common parent. You will cross with different founder parents. Okay, different inbreds. Then after that, again you will follow single seed descent method. Then at the end of the day, you will get population. Okay. What you should remember here, in multi-parent population, you will use more number of parents. But in case of your biparental mapping population, you will start with only 2 parents. ठीक है, समझ गए ना? बताओ, कमेंट में बताओ जल्दी. Clear? Is it clear regarding biparental and multi-parent populations? Yes, sir. Yes, sir. Yes, sir. ठीक है. We will move to next question. In self-pollinated crops, what is the most important step to make sure the seeds produced stay genetically pure and true to type? In self-pollinated crop plants. Crystal clear. Shinchan. Arey bye. Thank you. B. Keeping the seed field isolated and using seeds from only one uniform plant type. That's what we will do in case of your pure line selection, in case of pedigree selection. We are only selecting the single plant till the F6, F7 generation. Then we will go for multiplication. Okay, and also we will go for field isolation. Okay, yeah, good, Nutan, good. So, Vasundhara Lakki, Nutan, Preeti, Ankit Kumar, सब लोग भी बता रहे हैं. Good, everyone. Letting different varieties of the same crop grow side by side, then it will do contamination. Mixing seeds from different varieties to increase diversity. अरे भाई हमको diversity नहीं चाहिए. In variety we don't need diversity, we need uniformity. Okay, if you maintain the diversity, then farmer will come. Then you will be out of your post. Okay, using machines to harvest the crop and cleaning the seeds after harvesting. Okay, in case of the breeder seed production, nucleus seed production, if you use the harvesters or any combined harvester, then again there will be a contamination. Okay, there will be a mixture. This is also not an option. Okay, your second option is correct answer. In R.D. Weinberg population, okay, anyway it will comes in your genetics portion, but I have took this. If the frequency of allele R, that is P is 0.8, allele R is Q is 0.2, which genotype frequency is correct. Okay, if you find this question, there is no other techniques. Okay, only one technique, that is your P square plus 2PQ plus Q square. Okay, this is only the formula for everything. Okay, they are giving P value, they are giving Q value. You just have to do your P square will be 0.64, your 2 PQ value that is 0.8 into 0.2, okay, 0.16, ठीक है. उसके बाद 2.32 plus your Q square 0.2 into 0.2, 0.04. सही है क्या? Your Q square value, P square value, that is nothing but your P square capital R. 2 PQ is nothing but capital R, small r, small r, small r. बताओ कोई सही है क्या? सारे तीनों आंसर. 0.64, 0.32 कितना हो जाएगा? 0.64, 0.32, 4 2 6, 6 3 9, 0.96 प्लस 0.04. ठीक है. 09101 ठीक है, सही है. C. Okay, out of these they have given RR frequency, they are telling 0.4. No, it should be 0.04. Your capital RR frequency will be 0.64. That is also not correct. Your capital RR frequency they are giving 0.48. That is also not correct. They are giving capital R, small r frequency, that is 0.32. If they ask about RD Weinberg law, you should go for this formula. Okay, there is no need to waste your time. You will get an answer very quickly. Okay. Which of the following is not an assumption of the RD Weinberg law of equilibrium? If anyone asks you the definition of RD Weinberg law, you will start with, okay, you should be start with in large random mating population. Okay, the principle itself starts with in large random mating population. Your gene and genotypic frequency will remain constant of generation after generation. Okay, when there is an absence of mutation, migration, random drift, mutation, migration, random drift, selection. Okay, when there is no of these and in large random mating. Okay, your definition will itself suggest that in large random mating population, your gene and genotypic frequency will remain constant generation after generation when there is no selection, mutation, migration and random drift. Okay, this is the definition, not an assumption. Mutation is an assumption, random mating is an assumption, your selection is also your assumption. Small population size, it's not an assumption. Okay, C. As I told, in large random mating population, this is your assumption. Okay, sir please marathon for all subjects. अरे भाई मैं ये बस एक ही सब्जेक्ट ये कर सकते हैं, सबको सारे चल रहे हैं ना, वही you're asking. There are already in each and every subject there will be a marathon. Okay, already about to complete in different, in case of social science, in entomology also it started, in agronomy वो भी चल रहा है. ठीक है, पैथोलॉजी तो हमारा कंप्लीट हो गया. Okay, we are doing for every subject, don't worry. Genotypic frequency remains constant generation after generation until there is no mutation, migration, random drift, selection. Good, that's what I told. But Sinchan, when you are telling the definition, okay, when you will come to your MSc, if you are doing genetics, okay, when you are Viva, when you are someone examiner will ask you, you should start with in large random mating population. That is the best, the first assumption. Okay, yes, good. अभी आ गया देखो. From this question onwards, you will get so many questions based on the heterosis breeding. Along with the dominance and over-dominance hypothesis, there is another hypothesis called epistatic hypothesis of heterosis. The following are statements about epistatic hypothesis of heterosis. Select the correct statement about the epistatic hypothesis. सर IRI आऊंगा तो ट्रीट दोगे क्या? अंशुल टेकम अरे भाई आपका नाम तो मैं याद में रखेंगे, ठीक है? अंशुल, आप आ जाओ IRI, उसके बाद ले लो क्या पार्टी चाहिए आपको, ठीक है? बस ट्रीट नहीं देंगे, आपको क्या चाहिए वो देंगे, आ जाओ मेरा दोस्त। इसको आंसर बताओ अभी। बी 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 okay, ABD only, ABD only. You can easily say epistatis is also known as non-allelic interaction. Yes, absolutely. Okay, your epistatis is nothing but interaction between two genes. Interaction between two genes means those are non-allelic in nature. Heterosis has positive association with the presence of magnitude of non-allelic interaction. That's what your epistatic hypothesis will tells. Okay, आपका epistatic hypothesis क्या बोलता है? ठीक है, वो बोलता है आपका heterosis is due to epistatis. The non-allelic interaction are two types: additive into additive, dominance into dominance. Then what about your additive into dominance? ये भी आपका epistatis है ना, वो मिस कर रहा है वो थर्ड स्टेटमेंट में, ठीक है? Only they are telling A into A, D into D. What about A into D? That's also your epistatis. Okay, this is not an option. Epistatis, particularly those that involves dominance into dominance interaction, may contribute to heterosis. Yes, your epistatic hypothesis will explain. Out of three interaction, okay, the dominance into dominance, you know, the major contribution to your heterosis. That's what it will tell. Your answer is, you should neglect C, so that your answer will be A, B, D only. Yes, good. Preeti, A into D, good. Shinchan, good, good. Okay, before we are entering into the next questions, okay, first we should know these two important hypotheses. That is your dominance hypothesis and over dominance hypothesis. Proposed by Davenport, okay, in 1908, and it is explained and elaborated by Bruce and Keeble and Pello. Okay, you should keep in your mind, okay, Davenport, okay, who gave the particular hypothesis. And over dominance hypothesis, that is by Shull and East. Okay, what is the basic idea behind these two hypotheses? Okay, favorable allele dominant alleles from one parent mask the deleterious recessive allele from the other parent. Okay, the heterosis is due to masking effect of your recessive allele. Okay, not due to heterozygosity. Okay, it will due to the masking of this particular allele with the dominant allele. But in case of your over dominance hypothesis, your heterozygous genotype itself is superior to either homozygotes. Okay, your heterozygosity itself is superior. But in this case, your dominant allele is suppressing your recessive allele. That's why you are getting heterosis. So that's why, due to that idea, you can see capital A and capital A small a, both are similar. Okay, in this capital A small a, your capital A is suppressing that small a and it is giving heterosis. But in case of capital A, there is no suppressing, there is no need of suppressing. Okay, so itself it will give heterosis. So both are equal compared to small a. Okay, your capital A and capital A small a. But in case of this particular condition, heterozygosity itself is the reason for over dominance hypothesis. Your capital A is superior over both capital A and small a. You can see here, your capital A is superior over capital A and also it is superior over small a. Okay, clear? Cause of heterosis, masking of harmful recessive gene, but here the advantage of heterozygosity per se, your heterozygosity loci itself. Your dominance hypothesis will tells it is the heterosis directly proportional to number of dominant genes. Number of dominant genes, whether maybe it is in homozygous condition or heterozygous condition, we are not worried about it. It is directly proportional to number of dominant genes. But in this case, in over-dominance hypothesis, it is directly proportional to number of heterozygous loci. Okay, your loci should be heterozygous here. But in this case, we are not worried about heterozygous. It should be dominant. Okay, in case of dominant, you can see dominant homozygote and dominant heterozygotes. Both will the reason for the heterosis. Example, okay, as I told in the particular basic idea, okay, if you cross any two parents with a diverse, when they will comes in heterozygous condition or in homozygous dominant condition, you will get an heterosis. But in this case, at particular loci, okay, it should be heterozygous in nature. Prediction: if all favorable dominant genes are fixed, heterosis can be recovered in a pure line. You can expect the heterosis in inbred line also. Okay, the perfect inbred line. Okay, where all the genes, okay, are having dominant homozygotes. Okay, this is the prediction in case of a dominant hypothesis. But in case of over-dominance hypothesis, it should be only in heterozygous condition. Okay, that's why we cannot fix the heterosis in case of over-dominance. It should be in heterozygous condition only. Okay, if you go for fixing, that means if you go for selfing, then you will not get an heterosis. But here, as per dominance hypothesis, as I told, it is directly proportional to number of dominant genes. We are not worried about the condition. Maybe it is in dominant homozygote or dominant heterozygote. So you can go for fixing also. Okay, if your inbred is completely homozygous for all the genes, so you can expect the heterosis. Again, there will be a number of objections to that particular dominance hypothesis. We will see in the marathon classes if time permits. At this point of time, you should know what are the basis about dominance and over-dominance hypothesis. If they ask in your exam, which is the most accepted hypothesis? बताओ, comment में बताओ, which is the most accepted hypothesis of your heterosis? ये तो clear है ना? The difference between dominance hypothesis and over-dominance hypothesis. How they are trying to explain the concept. Dominant. Yes, good. Okay, your dominance hypothesis is the most accepted. Okay, and again, if I want to tell genuinely, at this particular era, okay, at this particular time, if anyone asks you, okay, the best answer should be it is a cumulative effect of dominance, over-dominance, and epistatic hypothesis. Okay. Now, at this particular era, now we have understood the different hypotheses and different phenomenon. Okay, now we can say that it is a cumulative effect of dominance, over-dominance, and epistatic hypothesis. But if they ask you in exam, if they are giving the 4 options, then you can choose the best accepted is dominance hypothesis. शिंचन ओवर डोमिनेंस बता रहा है, क्या हो गया आपको? मैं बता रहा है ना, खुद में बता रहा है वो डोमिनेंट हाइपोथेसिस. Okay, then why you are telling it is over dominance? Yes, good. How we will support, why we will support dominant hypothesis explains much of the observed heterosis in the crops, your dominance hypothesis. But in case of over dominance hypothesis, it explains where only heterozygous will outperform the both the parents. At that particular condition only, we can support over dominance hypothesis. Okay, but in all other cases, it will be dominance hypothesis. Okay, just small information about our courses. Okay, anyway, there is a price drop. You can purchase our course for this particular exam. Otherwise, if you are preparing for next year exams, okay, the 2027, you will get our mock test series bundle, that is for 799, and our courses 499 for next year, that is for 2027. For 2028 also we have launched our courses, that is for 899. Okay, just go and enroll our courses. You can also take feedback from your seniors who are already purchased this course and they are getting the benefits. Okay, just get a feedback and just go and enroll. Don't waste your time so that you will miss the informations. So next question, AFLP combines restriction enzyme digestion with selective PCR amplification of the genomic fragments. The second statement, AFLP typically function as codominant marker system, providing easy heterozygote detection. अरे बताओ, अभी बताओ, ये तो मैं बार बार ऑलरेडी बोल चुका है, ठीक है? I have also told that the different markers and their dominance and okay, dominant or codominant. I have already explained each and everything. See. Okay, it is true. Your AFLP combines both restriction digestion then selective PCR amplification. ये तो मैं clearly explain कर दिया class में, ठीक है, markers में. AFLPs typically function as dominant again. Okay, it is not an codominant marker. Okay, your statement 1 is true and statement 2 is false. You can see, just I will give some glimpse how your AFLP will work. This is your genomic DNA. Then you will use two different restriction enzymes to cut your DNA fragments. Then you will select one of the fragment which is very suitable for you. Then you will use the adapters. Adapters are nothing but you know the sequence information. You will just use those adapters to the fragments end so that you can amplify that particular product. Since you know the end sequence that is from your adapters, okay, you can use the primers. Then you will, out of these three different fragments after your digestion, you only need to selective amplification of particular products. Then you will use some selective nucleotides in your primers, PCR primers. Then you will use the selective amplification so that you can reduce the complexity. Okay, that's why it is restriction enzyme digestion plus selective amplification. Okay, and also this is dominant marker where you can't differentiate between homozygous dominant and heterozygous dominant, but sometimes codominant. अरे मतलब कविता, there is no need to confusion here. If they ask in case of your exam, okay, you should choose it is dominant. Sometimes, okay, but sometimes there is no need to think like that. They will not ask you in this particular exams. Okay, we can convert all dominant marker into codominant marker if I want to say by using some advanced different genotyping methods. Okay, that's not a case, but the basic idea, the basic principle, it should be dominant. Okay, clear? RAPD, AFLP dot dominated by ISSR or dominated by RSSECC is the trick. क्या ट्रिक बता रहा है नूतन, मुझे समझ नहीं आ रहा है. राडी, RAPD, AFLP, डार्ट और डोमिनेंट. अरे उतना क्यों सोच रहा है, मैं तो एक्सप्लेन कर दिया ना बेसिक कॉन्सेप्ट, क्यों डोमिनेंट है, क्यों कोडोमिनेंट है. नूतन, आपको तो हमारा कोर्स में है ना. अरे आप कॉन्सेप्ट वाइज जाना पड़ेगा. वही विल make it very complex. If you know the basic thing, you can just easily differentiate between dominant and codominant marker. Why we are calling it. Again, if you want to remember, okay, we just there should be a your AFLP, RAPD, ISSR. These are your dominant marker. Okay, if you want to keep remember. Okay, there is no need to confusion. Okay, there is no need to think too much about it. Heterosis breeding involves crossing two genetically diverse inbred lines to produce F1 hybrids with superior traits. Your statement 2 is telling the vigor observed in F1 hybrids remains unchanged for many subsequent generations if the hybrid is self-pollinated. बताओ आंसर. See. Statement 1 is true and statement 2 is false. Your F1 hybrid, the vigor observed in F1 hybrid, okay, we can't make it what we can say permanent. Okay, it is changed in the subsequent generation. Reason also I have already told. Okay, your segregation. Okay, your capital A, small a will segregate in the further generation if it is self-planted crop. Okay, in case of cross-planted crop also, your purity will going to deteriorate since there will be a open pollination. Okay, whether it may be selfing or maybe open pollination, your hybrid vigor will get reduced after your F1. Segregation and recombination. Yes, good. Shinchan. Again, heterosis ka question. Heterosis typically manifest as the superior performance of F1 hybrids in the traits like yield, growth, and stress tolerance compared to homozygous parental lines. Yes. Okay. Superior performance of your F1 plants is nothing but your heterosis. B. The dominance hypothesis explains the heterosis by suggesting that deleterious recessive alleles from one parent are masked by dominant favorable alleles from the other parent in the hybrid. That's what I explained in the previous slides. Over-dominance hypothesis states that heterozygotes have a fitness advantage superior to either of the homozygotes due to allelic interaction. Yes, exactly correct. D. Narrow sense heritability is consistently higher in F1 hybrids, exhibiting strong heterosis, reflecting the increased additive genetic variance contributed by the parental lines. This is completely wrong. Okay? Your answer should be A, B, C only. Whatever you are getting in F1 hybrids, it is due to dominant genetic variance, dominance genetic variance, not due to additive genetic variance. Okay? It is due to dominance genetic variance. That's what your heterosis will tells, na? Whether it is due to the heterozygosity or the deleterious allele masking effect. There should be a dominance variance, not additive variance. Though, so this is the wrong option. They are telling narrow sense heritability is consistently higher in F1 hybrids. Okay, exhibiting strong heterosis, reflecting the increased additive genetic variance. It is due to increased dominance genetic variance. Okay, only A, B, C are correct. According to the over dominance hypothesis, the heterosis is the result of the superiority of the heterozygotes over its two homozygous parents. See, the same statements again. The principle is same. The slides I have showed the difference between dominance and over dominance. Based on that, they have asked 3-4 questions. Reason: the over dominance hypothesis states that the superiority of the heterozygous arises due to the masking of the deleterious effects of the recessive alleles by their corresponding dominant alleles. The reason they are telling like this. C. A is correct, but R is not correct. Yes, it is due to result of the superior heterozygotes over its two homozygotes. In reason they are telling, it states that superiority of the heterozygote arises due to the masking effect of deleterious genes. That is not over dominance. That is due to dominance hypothesis. The second statement. Okay, that's why only A is correct. Next question. Inbreeding depression is the most common in case of cross-planted crop plants. Statement 2. Repeated selfing, though it reduces the vigor, is necessary to create genetically stable inbreed lines that maximize heterosis in the F1. Batao, answer batao. Inbreeding depression is most common in cross-planted crop plants. Repeated selfing, though it reduces the vigor, is necessary to create genetically stable inbreed lines that maximize the heterosis in F1. Yes, good. A. Both statements are true. You can see, inbreeding depression is nothing but exposing of your recessive alleles. Okay. It is common in cross-planted. What about cross-planted crop plants? Your cross-planted crop plants will be in heterozygous conditions. Okay, they love heterozygosity. Your CP crops, okay, they love heterozygosity. That's why they will be in the heterozygous condition. Okay, due to the outcrossing or due to the cross-mated, due to cross-pollination. But you can see the recessive allele will mask by the capital A allele in natural condition. But you are inbreeding, that is nothing but you are forcing the self-pollination. Okay, when you will go for inbreeding, your small a will expose. So they will die, or they are the reason for depression, inbreeding depression in case of fitness, in case of reproduction, in case of yield. Okay, that you can see in cross-pollinated crop plants. But in case of self-pollinated crop plants, they love homozygosity. Okay, whatever the deleterious recessive alleles, okay, they will already exposed to the environment in earlier case also, in earlier case itself. Okay, after that evolution and generation, if they are having any recessive alleles, okay, they are not deleterious. Okay, they will already get fixed in the population. Okay, that's why you will not see inbreeding depression in case of self-planted crop plants. Okay, they love homozygosity. Repeated selfing, though it reduces vigor, is necessary to create genetically stable inbreed lines. Yes, you should go for inbreeding so that you can get an homozygous individuals as an inbreeds, so that when you will cross both the homozygous individuals, you will get an stable or maximized heterosis. Okay, both statements are true. Yes, remains want to be in the heterozygous condition by selfing deleterious genes export to in the cross-planted crop plants. Yes, good example, good Shinchan. Again, dominance hypothesis was originally proposed by Davenport in 1908, as I told. According to this hypothesis, heterosis result from the heterozygosity. It states that heterosis is directly proportional to the number of dominant genes. That's what I told. The number of dominant genes, whether it may be dominant homozygous or dominant heterozygous, we will not care about it. The number of dominant genes. It is correct. It is correct. In the heterozygote state, the deleterious effects of recessive alleles are masked by their dominant alleles. Again, it is correct. But it is not a result of heterozygosity itself. Okay, it is what it will. It is due to overdominance hypothesis. Will tells the you will explain this particular statement. So you just have to ignore this second statement. ACD. ACD. This is your correct option. Okay, you can see at each papers. Okay, from 2020, they have targeted any one of the particular portion. In this paper, they have targeted heterosis. That's what I wanted to tell. Again, match the following regarding different markers: RFLP, RAPD, AFLP, SSR. बताओ आंसर. आंसर लगा दो, जल्दी लगा दो. ये first we will see RFLP. Anyway, we already discussed about it. Combined restriction with selective Fisher amplification, that is your AFLP. Okay, uses short arbitrary primers under low stringency PCR. You will use short arbitrary primers 10 to as generally 10 nucleotides decamer. Okay, where you will use in random amplified polymorphism. Okay, in polymorphism detected by restriction enzymes and southern blot, that is your RFLP. Tandem repeats are nothing but your SSR. A2, B1, C4, ठीक है, D3, C. ऑप्शन इज C. अरे ये लगा रहा है क्या सारे? मैं कुछ गड़बड़ किया है क्या? फॉर ए सेकंड ऑप्शन, फॉर बी फर्स्ट ऑप्शन. ओके, सॉरी. For B, first option. For A, second option. Yes, that is your B. Array. Okay. C, fourth option. Sorry guys, sorry. A is your answer. Okay. Before that, you should know the basic principle. These are the basic principles of your different markers. Anyway, I already discussed in the classes. Okay. If time permits, we will discuss it in the marathon. Okay, why the different things for different principles. Which of the following statements about the scope of the IT PG RFA are correct? ये तो मैं बार-बार बता दिया IT PG RFA में. ठीक है, it covers the crop species that are listed in Annex 1. I have already showed that in the previous classes what exactly the Annex 1. It includes all the world's plant species across the every biome. ये तो मुझे पता नहीं है, अभी देखेंगे. It emphasizes food security as the key motivation for its coverage. Yes, it aims to cover animals that support agriculture. करते हैं क्या animals को? And it includes all world plant species. It will only covers the important, which are the crops which are important for food and agriculture. Okay, both are wrong answers. Okay, it covers only the some particular crops. That also I have told already, 64 crops. Okay, 35 plus 29, your food crops and forage crops. It will not cover all world food crops, and it will not also cover the animals that are in the agriculture. Okay, your answer should be A and C only. Okay, the D will be your answer. Yes, good. Arrange the following legal instruments in chronological order. अरे ये तो देखो रिसेंट में भी आ गया, ये तो पक्का आएगा. ठीक है, योर CBD 1993, PPFRA 2001, Biological Diversity Act 2002, International Treaty on Plant Genetic Resources, that is your 2004. Okay, you can see here now A, B, C, D. सही है, सीक्वेंस तो सही है सारे. यस, गुड. A, B, C, D. ठीक है, क्लियर. अरे इतना ही है आज का, मतलब इतना ही पूछा है प्लांट ब्रीडिंग से, और मैं थोड़ा IPR से भी लिया है, सीटेक का भी थोड़ा कुछ तो एक दो क्वेश्चन लिया था. ठीक है, दिस इज अबाउट थिंग इन दिस क्लास. ओके, व्हाट आई हैव एक्सप्लेन्ड, I explained clearly what about dominance and codominance hypothesis and your gammatic selection, that is for improvement, not for isolate. Okay, these are the different things we have discussed today. Okay, अभी बताओ, ये तो हो गया गेम ओवर. यस नूतन, गेम तो अभी PYQ का चला गया. अभी बताओ, मैराथन चाहिए? मैराथन चाहिए तो मैं ऑलरेडी बता दिया क्राइटेरिया क्या है मेरा. ठीक है, मैं तो ऑलरेडी दोनों बता दिया. मैराथन चाहिए, करो, क्या करते हैं आप लोग, सबको शेयर कर दो. सर डाउट, राइस वेराइटी पूसा DST1, DST जीन एडिटेड एट 3rd और 6th क्रोमोसोम. ये तो 6th क्रोमोसोम में था, मैं ऑलरेडी एक बार बताया था. मैं फिर से ना एक आपको पर्टिकुलर क्वेश्चन, ओके, आई विल डिस्कस इन मैराथन. ओके, अगेन दिस इज इंपॉर्टेंट एज आई टोल्ड. ओके, वी विल डिस्कस which is the gene they have edited and what is the regulation, negative or positive regulation of that particular gene. Okay, which is the variety and the mechanism, CRISPR-Cas9. We will discuss each and everything. Okay, I have also confused now 3rd or 6th. एक बार क्लास में तो बताया था मैं 6th, ऐसे मैं एक बार चेक करके फिर से बता देंगे. Yes sir, yes sir. Marathon is must. Marathon is must. Okay sir, thank you so much. Yeah Sajan, I will definitely cover that one. Okay, sure sir, we do it for marathon. अरे मैराथन के लिए नहीं, अभी इस वीडियो के में आपको करना क्या पड़ेगा, शेयर करना पड़ेगा सबको. ठीक है, 2K व्यूज आना चाहिए, मतलब सब स्टूडेंट्स के लिए ये रीच होना चाहिए. उसके बाद आएगा मैं मैराथन में. उधर तक मुझे मत पूछना सर मैराथन कब आएगा, मैराथन कब आएगा. ठीक है, ओके देन इफ इट इज the thing, if there are no doubts, okay, will if it reaches then we will see in the marathon. ठीक है, मतलब वो पता नहीं, मतलब देखेंगे 4-5 दिन के बाद. उसके पहले सारे जेनेटिक्स का वो शुरू हो जाएगा, वो सारे कवर कर दो जेनेटिक्स और सीटेक का. अब मैराथन में सारे. इन इंट्रोडक्शन क्लास यू सेड 6th, इन स्ट्रेस ब्रीडिंग यू सेड 3rd. okay, I will definitely clarify your doubt in the marathon class. East or West, Abhishek Sir is best mentor for plant breeding. Thank you so much for, अरे भाई thank you. कमेंट्स तो लगा रहा है, मतलब थोड़ा व्यूज भी आना चाहिए ना, वो भी थोड़ा मजा आएगा. ओके देन टिल दैट गुड नाइट फॉर ईच एंड एवरीवन. ठीक है, प्रिपरेशन में लगे रहो. ओके, विल डेफिनेटली गेट गुड रैंक. ओके, इन योर एग्जाम्स. ऑल द बेस्ट.